All right, so this video's topic is, probably one of the most important ones for our studies of muscles and nerves. It's actually going to be relevant to both, and so it's something that you really need to make sure that you understand well, and that's going to be the action potential. Some cells, electrically excitable cells like muscles and neurons, have the ability to conduct an action potential down their membranes. Um, so let's actually take a look here and start about how the membrane gets electrically charged in the first place. So if you'll recall from early on in the class, we talked about symporters and antiporters and primary versus secondary active transport. Well, pretty much every cell in the body has um, proteins in it called sodium-potassium pumps. These are sometimes called sodium-potassium ATPase pumps because they are an active transport mechanism. And these are operating in pretty much every cell in your body all the time, but they are particularly abundant and particularly important in nerve and muscle cells. And these proteins do something that's pretty simple. They bind to and use ATP, and they bind to 3 sodiums on the inside of the membrane. They then use the energy, hydrolyze the energy of that ATP, turning it to ADP, and that moves the sodium to the other side, allowing that sodium to leave, and it opens up a binding pocket for 2 potassiums on the outside of the membrane. Those 2 potassiums bind, as you see here, and then the phosphate from the ATP resets, and the enzyme moves those potassiums inside. So this is an active transport antiporter. This is a direct active transport antiporter that moves 3 sodiums out and 2 potassiums in for every ATP spent. And there's tons of this thing in the cell that is constantly working. This has a few consequences to it. The first consequence is that you set up an electrochemical gradient. So an electrochemical gradient is a gradient that has both a diffusion, that's the chemical, and a electrical charge separation portion to it. So we've got all of this sodium, potassium, ATPase in the cell. It has been moving all of this sodium outside the cell, and it's been moving all of this potassium inside of the cell. And so there's a chemical gradient, right? Which way does diffusion say that the sodium would want to go? Well, sodium would want to diffuse this way, and potassium would want to diffuse that way. Things want to diffuse down their concentration gradient, but they can't because the membrane does not let them through. Well, or at least the sodium can't because the membrane doesn't let it through. The membrane is actually more permeable to potassium. Sodium can only go through the membrane if there are special sodium channels that allow it to go through. These are usually going to be gated channels that can open and close. The membrane typically has potassium channels in it all the time. A limited number of them, but potassium can move through the membrane back and forth somewhat at will. So that is the chemical portion of it. You can probably understand, oh yeah, this is a simple diffusion gradient. I know how diffusion works. Here's the electro portion of it. Remember that we would move 3 sodium out. For every 2 potassium in. That means that every time we spend an ATP, we have a net positive charge delivered out. And the outside becomes more positive, and the inside becomes more negative for every 1 ATP spent. Well, you've got hundreds of thousands of these enzymes in every cell that are constantly spending ATP to do this. This is actually a pretty reasonable percentage of your resting metabolism, like something like, I don't know the exact percentage off the top of my head, but I would not be surprised to find out that it was something like 20% of your resting metabolism. The amount of energy that you burn when you're just like not doing anything, you're sitting there on the couch or whatever, is probably due to this. Like, this is constantly happening in every cell in your body. They're always burning ATP. And that means that over time, over a pretty short amount of time, you build up a whole bunch of positive charges on the outside, because you're always moving 3 positive charges out and 2 in. So you always are moving more positive charge out than in. And so over time, you get the outside has a net positive charge, because you've been moving more positive charges out there, and the inside has a net negative charge. That means that there is an electric field. And that electric field is about like this. And it's across the membrane. So if we imagine the electric field going from positive to negative, it's pointing across the membrane like that. These sodiums don't just have a diffusion gradient that wants to push them across. They have an electrical field as well. They are not just wanting to go across because it is because they're concentrated here and it's less concentrated inside. There is a negative charge on the inside that's pulling them physically in, but they still can't get in because there's no open channels for them to get in. Now, potassium feels those 2 forces as well, but in the case of potassium, they're actually counter to each other. Potassium could diffuse across, and sometimes does, because of the chemical portion. The chemical portion is forcing potassium out, but the inside is negative, the outside is positive, so potassium is actually going to be attracted to the inside, and it's going to be repelled from the outside. So even though potassium can leave, under normal circumstances, it doesn't. So this is the force that your body harnesses for secondary active transport. Right? When most cells do secondary active transport, what they do is they have the sodium-potassium ATPase. That's the primary active transport, where they set up this electrochemical gradient. And they'll have a, let's say, a symporter that will bind a sodium on one side and something else that you want to bring in, say a glucose on the other side, and that sodium really wants to come inside. And so this symporter says, okay, sodium, you can only come inside if you also bring a glucose. And it is the force that was set up, the electrochemical gradient force. The sodium out here wants to move inside so much that it will drive the active transport of glucose inside as well. That's how this electrochemical gradient is used by most cells. As we will see, it can actually be used by electrically excitable cells, neurons and muscle cells, to do something very different and interesting. Now we're going to jump back a bit and talk about the neuromuscular junction. So last time we talked about the structure of the muscle fiber. What we talk about today is definitely going to be relevant to cardiac and smooth muscle. Cardiac and smooth muscle have action potentials as well. But we're going to talk about it in the context of what's happening in a skeletal muscle. And skeletal muscle is, quote unquote, voluntary. Which doesn't always mean that it's directed by your conscious mind, but it does mean that it's always directed by your nervous system in some way. Skeletal muscle cells don't contract and don't propagate unless they are told to by a nerve. And here you can see this is a skeletal muscle fiber here. This is a skeletal muscle fiber. This is a skeletal muscle fiber. And this, you see here in white, is an axon, is an axon, right? And as this axon approaches its target, it sends off what are called coterminals, or branches. And so like this axon sends off a branch here, and that branch ends at this fiber. It sends off a branch here, and that branch ends at this fiber. It sends off a branch. Let's see. I don't see a branch that's going to this muscle fiber up here. There may be one somewhere else, but I don't see it. When the signal comes down this neuron, right? So there's actually an action potential traveling down this neuron. It gets here, and that action potential is going to split. Action potentials follow membranes. So the membrane splits, so the action potential is going to split, and it's going to end at both of these neuromuscular junctions. Neuromuscular junction is where a neuron terminus or an axon terminus gets really close to a muscle cell. Hence neuromuscular junction. And they don't just get close. There's actually some connective tissue that are holding them together. And the two things are sort of like form-fitted together, almost like your hand and a glove, all right? So this axon termini gets really close to the muscle membrane, but there is a short gap. They don't actually touch. There's a short gap in between the axon terminus and the muscle membrane. Remember that the membrane of a muscle fiber is called the sarcolemma. So there is a short gap between the axon terminus and the sarcolemma, and that gap is called the synapse. And even though you had an electrical signal that was coming down here, it's going to have to take a chemical form to bridge that gap. The electrical field can't actually bridge that gap. We're going to have to get the release of chemicals that will carry the signal from the nerve to the muscle. Here you can see sort of a cartoon image of a neuromuscular junction. This here, this is the nerve axon that the signal is going to travel down. And at the end of that axon, the axon termini, is what's called the synaptic bulb. The nerve axon actually sort of like goes out into this disc type structure. And at the end of that disc, there are all of these synaptic vesicles. They're all gathered right here, really close to the membrane, ready for exocytosis. And when a signal comes down the axon and then propagates across this border here, it triggers all of these vesicles to release. And they make the chemical inside of them, which is typically acetylcholine in neuromuscular junctions, but is some sort of neurotransmitter, fills this synapse and carries the signal to the muscle cell. As you can see, the neuromuscular junction isn't just like the axon bumping up against the muscle cell. This is a sort of complicated structure. First off, there's this area here called the basal lamina that is a little area of basically connective tissue, which wraps around the top of the synaptic knob or the synaptic end bulb, and basically keeps it from pulling away from the muscle. So it's like kind of, it's glued in there. Secondly, where the synapse is, note that there's this infolding of the muscle, the sarcolemma, right there. And as you should know by now, whenever there is that sort of structure, that sort of foldy structure, that's there to increase surface area. And this surface here is going to be filled with receptors for that neurotransmitter, typically acetylcholine. And the synapse itself is full of an enzyme called acetylcholine esterase. Acetylcholine esterase is an enzyme that breaks down acetylcholine. This is very important because when your nerve stimulates a muscle, it needs to be like one sharp stimulus. You don't want it to turn on and then not be able to turn off. If that happened, your muscles would constantly be active, like you would just your brain would stimulate it once, and the muscle would clench and it would be unable to unclench, and that would cause a state of tetanus, what's called fused tetanus, which is when a muscle activates and then can't unactivate, and it's constantly contracting. All right, so I've got this really, really complicated picture here. And so I'm going to start off by explaining it, and then we're going to have a little animation where I show you exactly what's going on. So, this is a picture of a muscle fiber, and you remember the triads that we talked about in the muscle fiber video? Well, you can see some triads here. This this here is the sarcolemma, the basically cell membrane. Here is that infolding, right, where it's surrounding the nerve termini, the synaptic end bulb. I haven't drawn in all the squiggly lines because I'm not that awesome at PowerPoint, but you know that they would be there. Here you can see that the sarcolemma actually takes a dip inwards and caverns inwards, and so you should recall that that structure where there's like the sarcolemma actually dips in through the cell, that is the T tubule. And here you can see this cistern here. This is the terminal cistern of the sarcoplasmic reticulum. And then these here, these are the lines of the sarcoplasmic reticulum going out. There's also a terminal cistern over here, but I've drawn it sort of faded because I'm going to be drawing a lot of other stuff there as well. But over here you can see the full structure. Here we have the T tubule. And here we have the terminal cistern and the other terminal cistern, and these are the lines of the sarcoplasmic reticulum going out, and the terminal cistern of the sarcoplasmic reticulum is full of calcium. Now, there's sodium potassium ATPase in this molecule. You can see that's this signal up here. And so you can see that it's distributed throughout the membrane. We also have in our neuron, we have vesicles in here that contain the neurotransmitter acetylcholine. That's those black triangles. And we have various types of sodium and potassium channels, right? So these here are ligand-gated sodium channels. They only open when acetylcholine binds to them. These, and note that these ligand-gated sodium channels, they're only found in the synapse. They're not found in the rest of the membrane. These voltage-gated sodium channels here, they are held closed by the electrical field that is generated by the electrochemical gradient. So they're held closed so long as the electrical field is strong enough. And they're found all throughout the membrane. These red channels here, those are potassium channels. They're more leaky, but they can allow potassium to move in and out. And we have two more receptors that we haven't talked about yet. We will talk about, and those are DHP, or dihydropyridine, which you see these little pink rods here, like this, and rhianidine, which are these blue rods here. And you always find dihydropyridine, DHP, and rhianidine close together. You always find them in the triad. The DHP is always on the sarcolemma of the T tubule of the triad, and the rhianidine is always on the plasma membrane of the terminal cistern. Remember, these structures in this triad are really close together. So even though they're on, like one's on the T tubule and one's on the terminal cistern, they're really, really close together, and they can interact with each other. Rhianidine is a calcium transporter, and it can be open or closed. They're closed right now because we want to keep the calcium inside. Now this thing down here, right, this is a little graph of the electrical potential of the membrane. Just to remind you, electrical potential means how much charge separation there is. So how much, basically, how much charge difference is there between the outside of the cell and the inside of the cell? It measures the strength and direction of the electrical field. So let's set things up. First thing that happens, so what you just saw there was the sodium-potassium ATPase pump worked to move all of the potassium all of the sodium outside of the cell and all of the potassium inside of the cell. So right now the cell is reset. And down here, our membrane voltage is at -70. All right, that means that there's a -70 millivolt difference of charge between the outside and the inside. You don't need to know exactly what that means, but you need to know that -70 is the resting potential. Like if the cell's not undergoing an action potential, that's where it sits, right? That means that the electric field is basically pointing inside the cell. You have more positive charge outside and more negative charge inside, and the electric field is pointed inside the cell with a strength of 70. Negative means pointed inside. So we're going to actually put in those electric field lines. All right, so you can see here, these electric field lines, they're pointed inside the cell, and they have a strength of 70. Now, if you look down here, it says that -55, and that says threshold. That is where the voltage-gated ion channels will open. So a electric field strength of -70 is sufficient to keep the voltage-gated sodium channels closed. If that voltage ever gets up to -55, the channels will open, and then some cool stuff is going to happen. All right, so let's see what happens next. So we get an electrical signal down the neuron. When the electrical signal reaches the axon of the neuron, we released through exocytosis our acetylcholine, and the acetylcholine went into the synapse. The acetylcholine binds to the ligand-gated sodium channels, and those ligand-gated sodium channels become open. Not all of the sodium channels become open, just the ones in the synapse. So let's see what happens. Oh, and you can see here that our position, marked by that black dot, has advanced. We're still at -70 as far as our membrane potential because no ions have moved across. Okay, so since those sodium channels became open, sodium in the local area has moved across. That means that at least in this local area, the electric field has weakened. And now it's weakened to the threshold value because some sodium has moved across. When we hit that threshold value, again, this is a local phenomena, we're going to symbolize the fact that the electrical field has weakened by turning these arrows around. They're now no longer strong enough to keep ligand-gated ion channels closed, or to keep voltage-gated ion channels closed. And you look here, there aren't very many voltage-gated ion channels in the synapse, but there are some close to the synapse. So those channels are going to open up. When those channels open up, sodium flows through them. And when sodium flows through them, that's going to cause the electrical field near that area to weaken as well. And that causes more sodium gates to open. This is a chain reaction. Maybe as a kid you played games with dominoes where you set up dominoes in big long rows, and then you push the first one over and it knocks the second and the third and the fourth and the fifth, and it goes bang, and you can see like YouTube videos where people have done this with like millions of dominoes and things like that. That's basically what we're watching here, right? The first domino was this acetylcholine here letting some sodium in here. That tripped the next domino, which was the voltage-gated ion channels that are close by. They let sodium in them. And then they tripped the next domino down the road. And note that this is spreading in both directions. So let's see this. Okay, we've got sodium that's now moving in there. And that's going to change the electric field direction and strength in the next part of the membrane. And note that this follows the shape of the membrane. It doesn't just go left or right, it follows the membrane. So it's going to dip inside of this T-tubule and change the direction of the electric field across the membrane in this T-tubule. That's going to open the ligand-gated ion channels, or the voltage-gated ion channels in the T-tubule. And then sodium is going to rush into the cell in the T-tubule as well. Now at this point, you'll note that our voltage has been moving up, right? The more and more sodium that we let in, the less electric field strength we have. At this point, we've actually let so much sodium in that we've passed zero, and the electric field really has flipped. Now we've let so much sodium in that the inside of the cell has become positive, and the outside of the cell has become negative. This is what's called the overshoot. Now there's a few important things that are going to happen here. Let's take a look at them one at a time. First off, we shouldn't forget the synapse, right? The synapse has been being stimulated for a while. It's still letting sodium in through the synapse because there's still acetylcholine bound to these ligand-gated sodium channels. Some of the acetylcholine will be what's called reuptaken by the synapse itself, right? This is actually the most economical way to do things, is that the synapse just binds to the acetylcholine itself, takes that acetylcholine back in through endocytosis, and then it can use it again later. Some of the acetylcholine is going to be destroyed by acetylcholinesterase. Remember, that's that enzyme that destroys acetylcholine. So those are the two ways we're going to get rid of acetylcholine. We have acetylcholinesterase that's going to just destroy acetylcholine. We also have a acetylcholine reuptake mechanism that's going to just like grab the acetylcholine and pull it back into the neuron. Well, now we're not stimulating these ligand-gated sodium channels anymore, so they close, and they now no longer allow more sodium to flow in. We also have something that can flow out. So remember, these potassium channels, they're pretty much leaky. They allow potassium to go in kind of whatever direction it wants. So now that the inside of the cell has become positive, all of this potassium says, well, wait a minute, I'm positive. I don't want to be in this whole positive area. And they start flowing outwards again, starting at the synapse. And then that reverses the electric field there, because you've got positive charge flowing out. And that closes the local voltage-gated sodium channels. And then potassium flows out. Oops. Potassium continues flowing out of the rest of the cell. But note what's happened here. This reversal, this reversal of the electric field here has pushed our dihydropyridine out. And when the dihydropyridine is pushed out, it moves, it opens the ryanodine. And when this ryanodine is open, that allows calcium to flow out. And at this point, we've had a lot of potassium that's been flowing outside of the cell. So more potassium flows out, and that's going to cause all of the rest of the voltage-gated ion channels to close. And at this point, so much potassium has actually left that we've not only got back down to -70, we've overshot that -70. And we're down to like -90 or something like that. During this time, this is what we call the so during this time, when the membrane is overpolarized, the nerve can't, or the muscle can't be stimulated again. This is called the refractory period. The nerve can't be re-stimulated until it basically resets itself. And what's going to reset it? Sodium-potassium ATPase. Sodium-potassium ATPase has been at work constantly in the background and is still chucking sodiums out and pulling potassiums in. And it then resets the resting potential back to -70. And once the resting potential gets back to -70, this muscle can be triggered again, and another action potential can be initiated. So some important things to keep in mind here. The action potential started at the synapse, and it spread out in both directions from the synapse. What the action potential was, what is actually spreading out, is the flipping electric fields. So what you get is the ions aren't moving left and right. The ions are just moving in or out. But when you have positive ions moving in, that triggers voltage-gated sodium channels to open, which causes more ions to move in, which causes more channels to open, which causes more ions to move in, which causes more channels to open, and like every channel that opens allows enough ions to flow in to open the next channel. That's those falling dominoes again. So this wave of switching electric fields, like weakening and then reversing to the opposite direction, that's what's flowing down and across the sarcolemma. That's what the action potential is. And here, when our acetylcholine stimulation stopped and the potassium started coming out, what we had was a wave of the electric fields flipping back. And so what we have is a temporary thing, right? You have electric fields flipping down, and that propagates across, and then you have electric fields flipping up, and that propagates across as well. So you just have this, like, if you've ever seen, like, the wave in a stadium, where you have this wave that goes across, like, the benches, and just people standing up and then sitting down. The people aren't actually moving. They aren't going from left to right. But you see a wave that moves from left to right because the people stand up and then they sit down. So that's what's happening here. The wave that's moving is the electric field. The ions are just moving to one side of the membrane and then the other side of the membrane. But they're doing so in this repeated fashion, like the people in the stadium. And that's what creates that wave action moving down our bleachers, which in this case is our sarcolemma. All right. So that's the action potential. To recap, you need to know what sodium potassium ATPase is and how it works generally. You need to know how sodium potassium ATPase establishes the electrochemical gradient and what that electrochemical gradient is. You need to know what a neuromuscular junction is. And you need to know, in general, the steps of an action potential. That you have a release of neurotransmitters that activates ligand-gated sodium channels. Sodium rushes in. That triggers voltage-gated sodium channels to let more sodium in, and that's what propagates across the membrane. You then have that reset. Potassium goes out, and that reestablishes the potential, closing those sodium channels in a wave going down the membrane. And something that we just sort of briefly touched on here, when the action potential hits dihydropyridine, DHP, it changes the shape of DHP, causes what's called a conformational change, which is just a fancy word for a shape change. But because DHP is located so close to ryanodine, which is in the terminal cisterns, that DHP changing also opens up ryanodine, which has let calcium out. So even though, like, our action potential has come and gone at this point, this calcium is still out. And it's actually the calcium that's going to cause the muscle to contract. All of this song and dance with the action potential has just been to coordinate the release of calcium. And in our next video, we're going to talk about what that calcium does and how it causes the cell to contract.